We are surrounded by plants. They are important for the absorption of carbon dioxide and release of oxygen through photosynthesis. They are also the primary producers in a trophic pyramid, providing energy to the consumers on the upper levels. Plants exist in the thermo range of -10 to 60 degrees Celsius due to freezing of water and denaturation of proteins. They usually specialize and adapt to a specific niche range to guarantee enzyme stability, optimum growth, and functioning. When the environment changes, plants have various behaviors to respond to the stimuli. An important behavior is the nyctinastic movement of leaves. It's a specialized structure in the petiole that allows plants to fold their leaves together. This behavior has been observed in the weed species Pendula, also known as the Easter Cassia, specifically found in the coastal environment of Smiths Lake. There is a lack of research on the behavior, so we were interested to find out whether light triggers the opening of the leaves or if they follow a circadian rhythm, which is a natural internal process that cycles through the day. Furthermore, we looked at whether different wavelengths of light impacted the sleeping behavior. The knowledge will give insights into new weed management strategies, as the species is native to South America and regarded as a significant environmental weed in New South Wales and Queensland. Its rapid growth rate and prolific fruit production outcompetes native species. It could also give insights on the function of developing such a behavior within a plant species. To test out whether light triggers the opening of leaves, we decided to cover the plants and prevent them from accessing sunlight. We used plastic vials and covered them with layers of black plastic. We also left vials uncovered as a control to ensure that the containers do not trigger the response. They were placed on small seedling plants for an hour, and results showed that light does trigger the opening of the leaves. The plants covered under the black vials folded, whilst the ones in the clear vials still had their leaves open. We then looked at whether the different wavelengths of light impacted the sleeping behavior. We used a similar technique to the last, except we added extra treatments and left the bottom of the vials uncovered and stuck transparent colored plastic discs of red, green, and yellow. The vials were placed on different seedling plants, and the behavior was observed at 20 minutes, then every 5 minutes until a total of 45 minutes was reached. The treatment was also tested at different times of the day, morning, noon, and afternoon, to observe if there was a latency response. Results show a latency response from all treatments in the morning, whilst the red wavelength throughout the day delayed the folding response of the leaves. This means that the folding behavior occurs without the presence of light and specific wavelengths. The morning latency may also be due to the short period of time that the leaves have been open for. Future research could look at temperature, sunlight, and carbon dioxide intake to see if they impact the length of time taken to respond. The finding could also suggest that exotic species such as the Easter Cassia have undergone adaptive evolution to respond to the different climate in Australia and hence become advantageous for climate change in the future. Lastly, the adaptive behavior could increase the survival of weed invasions and become more of a problem to manage and make it difficult to maintain the endemic diversity of coastal ecosystems.